just like to remind you that returning from doubting Doug to now going back to doubting Dawkins, uh, in that video we had looked at how DNA makes RNA, makes proteins fold, and then the proteins have to interact with each other. And we talked about the interactome. All of those things are grossly simplified for the sake of just being able to highlight their significance. And we discussed that just looking at those factors made life coming self-organizing from simple molecules impossible. That cells come from other cells, and that is without exception because of the vast complexity of the things we talked about right up to the interactome, which is the many, many million combination uh, interactions between the different proteins in the cell. But even that does not explain the whole picture because there is another entire level of information found in the cell. And that's highlighted in an article written, and it's available online, called Cracking the Sugar Code, Why the Glycome is the Next Big Thing in Health and Medicine. Because as we were talking about protein interactions, it turns out that on proteins, especially on the surface of the cell, they are coated in sugars. Now, these sugars are not just a coating to make things taste good. They're actually not for that at all. You could envision the cell being like a peach, and the fuzz of the peach being like sugars, except you would have to multiply the fuzz by 10 in order to get the actual relative number of sugars on the surface of the cell. And these sugars are not just there for decoration. They are like a barcode that identifies the cell so that the antibodies in us would lock on to these sugars and realize that this is part of self and would not attack it. The sugars also communicate with other cells, and this is very early on. They don't know how this entirely works yet, but they're beginning to investigate this. The sugars also can modify the behavior of the proteins themselves. So if we talk about the complexity of the interactome, which is the interactions between the proteins, now we can broaden that out to these sugars that attach themselves to the protein, can alter the protein's behavior. And even more amazing than that is that all of these little protrusions coming off the cell contain information. And there's actual proteins that read this information. So you could imagine the entire wall of the cell being covered with messages, like a giant message board that particular proteins can come and read. And so it might say, help, I'm in trouble, come to my aid. There may be sugars that say, I need to be put to death. I am becoming cancerous, which is a known part of how they work. And so this has become known largely through researching disease. So they've discovered that rheumatoid arthritis, for example, is a disease of these surface sugars that cause the body or the immune system in the body to no longer recognize these particular cells as self, and so they start attacking these cells and cause the pain of rheumatoid arthritis. It is also the cause of autoimmune pancreatitis, and the same function as takes place there. We also know that some viruses can lock onto these sugars and cause disease. That's how HIV attaches itself to our cells. It utilizes these surface sugars, as well as certain streptococcus uh, bacteria will mask themselves so that they cannot be attacked by the body's immune system by uh, mimicking the sugars on our cell surface. This is so significant that they have called the sugar code the third alphabet of life. So the first alphabet of life that we know of is the DNA. The second is the proteome, proteins that relay information, and now they're just discovering sugar alphabet, glycome. And this promises to be far more complex than the DNA because there are so many more ways in which sugar can um, relay information. There's the capacity, it's mind boggling. And that's also one of the reasons why it's further behind than the genome because it's just so complicated. Now, you may wonder, why am I talking about all this? It's because I want us to be able to wonder. When you have popular scientists coming in on YouTube and the TV saying, oh, we're just a blob of meat, some insignificant speck on an insignificant planet in an insignificant solar system, blah, blah, blah. You know what I'm talking about? I'm trying to show and demonstrate that the wonders of this world are not an accident. Just keep in mind that everything I've talked about in these last two videos taking place in a single cell. And if we were to expand that out to human beings, it becomes mind-boggling. There are 30 trillion cells in a typical human being. 30 trillion. So all this stuff that I've been talking about is taking place 30 trillion times. This is not climbing Mount Improbable. This is bibbidi-bobbidi-boo biology. When you come up with a problem and you can't solve it, bibbidi-bobbidi-boo. You appeal to some natural selection, some random process to solve all of your problems. But it's nothing more than a fairy tale, except it's in reverse. It's trying to make our world smaller. And I assure you that that is not the case. And that is also why I'm not worried about new discoveries in biology or new discoveries in science, because the more we learn, the more awesome our God is. Behold the wonder of God and his creation. Heavens declare the glory of God, and so do we. Don't be afraid to be in wonder. When you see something on TV, it shows some amazing thing that you see in nature. It's okay to be in awe. We ought to be. Because just because you can describe something does not mean you've explained it. God has made these things to cause us to look to him in wonder and awe of how majestic he is. May you wonder at God's wonders today.